So I'm going to start with just a bit of a genetics 101, and then John's actually going to dive into the juicy science for me. And there's been some really great work done over the last 4 years out of the GCFF program, and I think you'll find it pretty interesting. First up, I wanted to say to Jeff this morning that radiata pine has still got a lot of potential, and I am still very excited about it as well. Right, so I'm just going to run through really a little bit of genetics 101. So here we go. Hopefully. Yes. So I think some of you have seen this before, but really, why do we bother with genetics, and do we believe in it? And really, if we start talking about where you start in a breeding program, you start with plus tree selection out of your landrace. So you go to the forest and you say, what are the best trees in the forest? You gather them together, and ultimately you're looking at about 5% improvement over your landrace, unimproved. What you do then in bring it together in a main population, and over several generations you can expect to get around 30% genetic gain or improvement in your growth rates. Now RPBC has got an elite population, and we know and can possibly prove in the next year or two that we will get at least 15% above the main population. So compared with our landrace or original where we started, we're looking at about 45% improved growth rate for radiata pine. Now, this isn't entirely a fair comparison, because genomics is also about speed of delivery. But once we integrate genomics and look at how it can help us estimate our breeding values and our GEF+, then we can look at least 10% gain on top of that. And if we get really excited and try and match clones and CP to site, because now we've got the ability to do that through some of our remote sensing work, then we can really look at maybe 60%. And if we get even more excited and start doing some tweaking of the genes, then perhaps the future could be quite different. So just imagine that, right? What are you planting in your forests? And just imagine the potential that you could reach. And I think I just wanted to put these two guidelines here, because John's going to present some results on large plot genetic gain trials that actually shows that above unimproved we have got 30% genetic gain. But remember, this is from harvest, and this is at least a generation ago, so we can expect that what you're planting right now is at least 30% improved off unimproved, okay? So here's where your seed stand or your seed select might sit. Are you excited about that in terms of your productivity? Here's your GF19, it's probably roughly where it might sit. Are you excited? Selected families or CP, I'm excited. Clones, well, now we're talking, right? If you get the right clone on the right site, that is a significant lift to your productivity and profitability, okay? And there's the future, in my mind anyway. So I just wanted to run through what breeding was so that people could actually start thinking about some of our beautiful jargon. If we look at where we start with a mum and a dad that we really like the look of, right? So we cross those two selections. Then we say, okay, I'm not really sure what the mum and dad do, so we go and test all their babies or their progeny in well-replicated field trials. And the reason we do this, not only a large number of trees per site, but across a number of sites, is to get to the bottom of what are the variation that we're interested in to carry towards to the next generation. You have to do a lot of trees, and you have to test a lot of trees. So that takes probably at least 8 years in the field before we actually then go and say, okay, let's measure them now, analyse it, look at the variation, pull it apart and figure out what part of the variation is a certain part of the genetic variation that we call breeding values. And they're really exciting, folks. And then we say, look, what are the best breeding values? We make the selection based on the breeding values and then say, oh, okay, let's get them into our breeding orchard and make crosses. So every time we go through one of those cycles, it's called one generation. And normally, when we make the selections after the breeding values, that's the point where you take grafts and you put it into your seed orchards, or you say, I want to make those crosses again and put them into a clonal program. What is forward selection? Forward selection is just that we say, when we make that selection, we trust that breeding value and we're going to take that one individual forward. So we'll take it, climb it, get scions off the top of it, graft it up and establish it straight in a seed orchard. And this is where we're moving, because historically the Radiata Pine Breeding Company and even at Scion historically have been using what's called backward selection. And this is very interesting for you to understand what the difference is. So you do a normal cycle over here on the left-hand side, on the right-hand side, sorry, and you say, okay, I really like that tree, but I don't trust the number. So I'm going to take that tree, a copy of it, and then make babies of that tree and make sure all the babies are good. And then after that, I'll take that tree to deployment. So it means you have to do 2 cycles of testing. That's historically what we've done. So you can imagine the delay sometimes of historically getting the best stuff out into the field, because it takes you over 30 years. Suboptimal, right? So what is genomic selection for those who haven't really been exposed to it before? Really, this is widely in the literature, but it's quite simple concept. Is instead of doing the progeny testing, we can say, using these markers, I trust these markers to tell me that that's a good tree. Now, it's a difficult jump for foresters to say, oh, I can't see that tree, right? So I don't quite trust those markers. So we'll probably start doing some interim measures in genomics and saying, I trust those markers, but I'm just going to test to make sure that tree's all right. And then as soon as we start trusting that, we'll be able to move into the markers only selection, right? But the exciting thing about genomic selection is that it actually speeds the delivery more than anything. So instead of waiting for that 8-year cycle, you can actually, as soon as you've got needle material or even cell lines, you can extract the DNA, get the markers and say, yep, that's a good tree. So that means the speed to market of what you can get in your genetic improvement program is rapidly increased. And we postulate that we can get it down to 9 years. What is a genotype? Well, 2 trees, if you put a lot of, if you go and look at the different changes in your genome sequence, they're called markers. And 2 different trees have different changes, and that pattern is called a genotype. It's very simple. You don't need to worry about what's underneath it. It's just the molecular characterization of that tree. So I just wanted to run through some work that we've done, and it's a simulation program where we did look at not only looking at genomics and how we could possibly speed up the breeding cycle with genomics, but how we might look at deployment on top of genomics. Because remember, speed to market is, again, one of the most important things for genetic improvement programs. So we've clearly defined that the biggest advantage of genomics is going to be delivering the same genetic gain in roughly half the time. Okay? And we are about to publish a paper on this, and don't be frightened, but I wanted to put this in so that you could have a look at the complexities of how you actually do your breeding cycle, that you've got to spend years grafting, you've got to spend years collecting the pollen, you've got to spend years waiting for the progeny test. So don't worry about it now, but look at it later if you're interested. Okay? And then there's another impact that genomics will have immediately before we even start trusting the marker pattern. And that is, we have a number of what we call selection series that you probably recognise, for example, as a 268 series. So 268345. And then there's the 888 series and the 8870 series. And they're just examples. But some of these series are not linked well in the breeding program, and the connections that will be available to us using genomics means that we'll finally be able to bring those things together. And I'm going to use some jargon now. This will improve the relationship matrix. That just means I know this tree and how it's related to that tree over here. Now previously that's not 100% clear using only our information from the historical program. Genomics will help us understand that relationship and help us get better breeding values. So that's kind of an aside as well. In our simulation, and remember this is a simulation, we've also shown that compared with forward selection, it's actually the higher heritable traits that we should expect even more genetic gain from the program. So this is exciting. And it's particularly exciting as some of the wood properties are really expensive to measure, right? So if we could train our panel, our markers, to understand wood quality, then we can actually deliver more gain in wood properties to you faster. And that, I think that's something that we need to think about. Oops. And as Brent said, I think it's really exciting. We've talked about genomics for a large number of years and marker-aided selection, but actually we're going to start doing things operationally. And I think, you know, this is pretty much almost world leading, particularly in radiata pine. And I think you should be proud of that. And so we've developed the SNP chip, and I have to acknowledge here Natalie Graham, who spent a lot of time on phone calls to the States very early in the morning, and her commitment has meant that we've access to a SNP chip technology through a consortium that's really competitive in pricing. So that's going to allow us to do 20,000 trees in the next 2 years. And so that's, you know, pretty exciting stuff. And again, I wanted to go back to propagation and speed to market, because basically we've got these genetics, we now need to get them out as fast as we can. And so we're working together with forest growers on that. And I'm really pleased and excited to say that we wrote a partnership proposal around increasing the efficiency of tissue culture, using biological active components and also semi-automation through bioreactors. And that has now passed the concept stage, and now we're into full proposal writing. And I know that some of you are going to be involved in helping with us for that. So thank you very much. I'm really looking forward to getting that over the line. And that's me. Thank you.
Thanks, Heidi, for sort of setting up the scene. So what I'm going to talk about in the last little bit of this presentation is really, if you take this improved genetic material and as forest growers you plant it, what gain are you going to get at the end of the rotation? How much extra value do you create? And also, how should we manage this improved genetic material so that we don't undermine all the hard work of the geneticists, that we actually can realise that gain? So this work, it's been an effort for a lot of people, so I just want to acknowledge the team that has worked on this project or program over the last few years. It's also been a collaborative effort between the Radiata Pine Breeding Company and also the Growing Confidence in Forestry's Future program, which we're going to hear about after the break. One of the things we've been very fortunate to have in New Zealand is this network of large plot genetic gain trials. Geneticists generally use single tree plots, so basically one tree to evaluate many different genotypes in their breeding programs. That's good for getting the relative rankings, but then we grow these trees as stands, and so these large plot genetic gain trials are expensive, they take time to generate results, but many people in New Zealand had the foresight to put these trials in, so we're very lucky to have that legacy. So what these trials enable us to do is to have the evidence of how these trees perform when grown at the stand level. And many of the trials we have in New Zealand also have a silvicultural overlay, so at different stand densities. These trials are also very important for providing the data to model growth, yield and wood properties of genetically improved trees. So if we want to understand and do predictions and make value propositions for making the case to deploy genetically improved material, being able to do simulations and have our models able to do those is very important, and the data to do that comes from these trials, and I'll show a little bit that in a minute. And they also provide important demonstrations to people. There's nothing like going out into the forest and actually seeing, stand A, which has got seed lot, particular seed lot, and then stand B that's got an improved seed lot, how that's performing, and particularly when you see some of the traits like straightness and branching, the differences are very apparent. And as I mentioned, we've been very fortunate in New Zealand to have this network of trials that have been established on a rolling basis since 1978. So we've used these trials to quantify realised gain for growth, and we've got data that have come from really 3 series that have been installed. The first large plot series, the 1978 to 84 genetic gain trials, then the next series that came in were the 1987 to 91 silvicultural breeds trials. So they had a silvicultural overlay. So we had seed lots with different genetic ratings growing at different stand densities, everything from 100 stems per hectare back in when sort of agroforestry was of more interest, right up to sort of 600 stems per hectare, in some cases even 1,000 stems per hectare. And then a more recent series, 1992 to 1994, so it's actually a while ago now, where they're actually looking at specific breeds, so seed lots that were bred to have high and low density as well as high and low growth rates. You can see on the map that these trials were installed pretty much throughout the range of conditions that exist in New Zealand. We have 42 installations of these trials, and if you look at the whole thing, there's 63 unique seed lots in those trials with different levels of genetic improvement. So it's a pretty powerful data set. And by having large numbers of trials, we can take out the effects of anomalies at particular sites. We've lost a few along the way. There's been the odd dairy conversion here and there, but we still have a pretty good data set. And we've used these data to quantify the effect of seed lot genetic rating on site productivity. We wanted to look at how could we look at realised gain in a way that enabled us to put it into growth and yield models so that we could actually simulate for the regimes of interest that we had what the extra gain we could expect due to improved genetics. So we did this by quantifying genetic gain in terms of the 2 common productivity measures used for radiata pine: site index, which is just mean top height at age 20, and then 300 index, which is the mean annual increment of a stand at age 30 grown at 300 stems per hectare. So those are 2 common metrics. We can then use that to initialise growth models. And we have some good background information. We have the spatial variation in these indices. So essentially we could look at change in 300 index and site index per unit change in GF plus growth rating. We did that work back in 2015, and the results have been published. The work is in the algorithms are in Forecaster, but we were very fortunate to get extra funding from the Forest Growers Levy Trust to do end-of-rotation assessments of the remaining trials, because if you think back to when they were planted, 1987 to 92, log prices are good. Many of these trials were disappearing, so we had a small window of opportunity to do end-of-rotation assessment on those. So we've been able to do that. We've been able to update the analysis. So our early analysis, just because of the age of the trials, we only had data at age 25 from 10 trial installations. So we've been able to update this now, so we have age 25 data from 39 of the trial installations, and we also have data from older ages, 27 to 30, from 29 of the trials. So basically we've confirmed the earlier results, but actually the story gets a little bit better. The gains that we'd assessed earlier on are actually, the gains we've assessed now are slightly better than the ones we assessed earlier on. So we get about 1.67%, it's getting rather technical, increase in 300 index per unit increase in GF plus DBH rating. What that really means, and what I think you're really interested in, is how much extra volume do I get at harvest? Heidi pretty much stole my thunder in the previous few slides, but we're seeing about a 27% increase in harvest volume if you compare GF plus, call it 10, that's a stand select, with a GF plus 25, which was the kind of highest level of genetic improvement at the time. And if you take that through to stumpage revenue, it's about a 33% increase in stumpage. So you're getting more volume, you're getting slightly larger trees, which means you get a little bit more of that volume in the larger, more valuable log grades. So you can see on the graph here, the relationship between your trait rating and the gain. So this is the gain in site index. You don't get so much gain in height, but you get the gain in 300 index in your essentially your basal area. And the key point is that the relationship actually gets steeper as the stand gets older. So the expression of genetic gain that you see at the start of the rotation isn't suppressed by competition at the end. In fact, it actually gets better. And that was always one of the concerns, is that people said, well, you might measure it early on, but will it be there at the end of the rotation? The answer is absolutely yes. We were also able to look at not just the gains that you get from increased growth, but actually genetic improvement is focused on improving form as well. So we're actually able to do pre-harvest inventories of these trials prior to harvest. And here's the results from 8 trials. So we were able to look at the volume by particular log grade. We could then put values on those log grades and look at how much total standing value we create in these trials. So this is standing value, this is not stumpage. Harvesting costs, transport costs are not included here. So this is just simply value on the hoof. So you can see, for a start, there's considerable variation among the trials. The more productive sites, you can see you get higher values. On some of the lower, less productive sites, you get lower value. In general, we know that stand density, site occupancy, is a major driver of value. So we can see that as stand density increases, we tend to get a fairly much of a linear increase. It doesn't hold across all sites, but in this case, so stand density was the major driver of value, but genetics still had an important role to play. And one of the key messages is, once you've taken that low-hanging fruit and lifted your stand density, further gains are going to come through through deploying improved genetic material. We also looked at relative value to try and take out that effect of stand density, and this is where we start to see the real importance of genetics, particularly the improvement in the stem form and branching traits. And this is work that Jonathan Dash, as Yaroslav and Heidi and I have worked on. So we can see that, again, there's quite a range of variation across the trials. The relationship now with stand density tends to be flat, in some cases maybe negative, but we can see that about 20% of the variation in relative value is due to the genetic differences among the seed lots, particularly the genetic differences with respect to branching and stem straightness. So that's growth. We've also looked at wood properties, and as Heidi mentioned, wood properties are highly heritable. So we've looked at 2 key wood properties, density, because we have a huge amount of information on density, and also stiffness, because this is a major determinant of the performance of structural timber. So in order to look at density, we had a national model of radiata pine wood density that had been previously developed, and essentially we used that model to understand density variation, so we could explain away the differences in age at measurement in the progeny trials, and then we can effectively look at what the genetic effect was. So we could put a genetic multiplier on this model using data from 679 families where we had core samples had been taken. The key point is that we found a strong relationship between the GF plus rating for wood density and what we've called this wood density index, which is just outer wood density of a 20-year-old tree. So again, we've tried to normalise that effect. So a 1 unit increase in GF plus density rating corresponds to about a 2 and a bit kilogram per cubic metre increase in wood density. So you can understand, if you need to get wood density above a certain threshold and you're on a particular site, you can look at what level of genetic improvement for density and you need to actually have to achieve your goal. We were fortunate to be able to do end-of-rotation assessments of the special purpose breeds trials, which, as I mentioned before, contained seed lots that were specifically bred to have high density and ones that also bred to have fast growth but had low density. So there were two series of these trials. There were the 1994 series, which had a wider range of seed lots in terms of genetic difference, and the 1992 series, which had a smaller range. If you look at the 1994 series, the results look really fantastic. The GF plus density rating, so low to high, and the outer wood density that we measured when we did the end of rotation assessments, there's a nice linear relationship. In fact, the slope's even higher than in our model. It's about 2.7 kilograms per cubic metre for every unit increase in GF plus density rating. We need to be honest though, when we did the 1992 trials, we didn't see that same effect, and we're interested to what's going on there. And I think some of the genomic technologies that Heidi mentioned can be used to untangle what's going on with this low density seed lot. We don't think that it's actually what's supposed to be in there is actually what is in there. The 1994 series was a single parent cross. This is a mixture of 16 parents, and we think there's some contamination has got in there. So we need to do a bit more digging to find that out. But hopefully the results, certainly on the top line, would convince you that the genetic gain for wood density is real. And it should be. The heritability of this trait is high. We've done a very similar thing with wood stiffness. Firstly, we had to develop a model for wood stiffness, so to be able to tease out the background noise. So we found that we can model wood stiffness as a function of age, temperature, and also the instrument that we use. So you can measure wood stiffness using different instruments, ST300, IML hammer, tree tap, etc. If we did all that, and we actually had data from both Australia and New Zealand, we can model temperature, so we can see that the acoustic velocity, which is a surrogate for stiffness, increases by about 100 metres per second for every 1 degree increase in temperature. Once we take account for that, we can then go and look at our progeny trials, which were assessed using a tree tap at a relatively young age. We can convert that to a normalised 20-year proxy for stiffness, the ST300 tool, and we can see there is no GF plus rating for wood stiffness, so we're using breeding values that a 1 unit change in breeding value corresponds to about 135 metre per second change in the age 20 ST300. So what does that all mean? Basically, it means if you're trying to grow for a particular target stiffness threshold, you can then understand how your distribution of log sonics will be affected by the genetic material that you plant. So if you're in this example here, say you're trying to target 3,000 metres per second, 3 kilometres per second, when you've got a breeding value of 0, so effectively you're on the average, you get about 20% of your logs getting better than that threshold. If you go to a breeding value of 1.5, so you go to a higher, very high stiffness rated seed lot, suddenly you're getting 50% of your logs over that threshold. And if you're supplying to a structural sawmill or an LVL mill where stiffness is key, that is a very material gain. So you can begin to do these simulations, these what-ifs, if you're needing to target particular customer requirements. Okay, so last slide, perfect timing. In terms of the outcomes, so really what these trials have enabled us to do is to quantify genetic gain and volume in a way that we can incorporate it into growth models, and through developing yield tables, you can incorporate that into forest valuations. We can do something similar for wood density. We have models that account for site silviculture and genetics, and we can show the extent of change in wood density that can be achieved by planting seed lots with particular GF plus density ratings. We've now done something similar for stiffness. And an important point to make right at the end is that that's the previous generation of large plot genetic gain trials that have told us something about the material that's in the ground is going to be harvested shortly. What about the next generation? And I think there are trials that have been established. It's really important that we continue to invest in those trials, because they will generate the results in the future. They'll generate that information that will help us model better model and better manage the next generation of genetic material to get the best from it. Thank you.